Okay, so hello everyone. Thank you, the organizing committee, for having me here. I will quickly go through my presentation. Thank you, Emilio, for the kind introduction. I'm Mark Anteghini. I'm working as a postdoc at Emilio's group, the Biofold unit, the University of Bologna. And what's the overview of our project? Yes, as many projects, we always started with a question that might be interesting for all of the audience here, which is how can we better understand the impact of genetic variants on proteins, what's already known and available, and how can we integrate it? Can we also take advantage of the recent developments in artificial intelligence, specifically protein language models? And how can we make our tool interpretable? What I'm going to talk about today is actually a part of a bigger picture. Our idea is to build mutation analysis platform that would merge different tools and scores and allow the user to get some level of explainability on the scores and the impact of mutation. As I said, we are going to integrate several tools. Among those, our effort is towards integrating protein language models in the equation. And finally, our scientific work packages conclude with the validation through real-world application. But today I'm going to talk about this central topic, the integration of protein language models, which were already mentioned in the previous talk. So what a protein language model does in very simple word is to learn compact vector representation of protein sequences that enable them to reconstruct their original input. In this talk, I'm going to show you the model we used in our predictions. This is the ESMC model that came out after the ESM2, which is son of the ESM3, basically. A lot of acronyms, but they always come with the ESM family published by Meta. And it comes with several versions, so increasing the number of parameters, also increase the number of dimension of the vector representing either residue or the protein. And what is usually done when representing an entire protein sequence, mostly for computational limits, is to aggregate the final layer of the model and come up with a single vector that represents the protein. Which is usually done by mean pooling. And in our study, we use the per residue representation, and we use the mean pooling for the full protein representation. This is a quick overview of the data set we looked into. We started looking into this data set published by a colleague of us, actually from university, from the Biocom group. Sorry for not putting the reference here. Sorry, Matteo, for that. I just noticed. But yeah, we kindly stole their data set, which is a combination of clean VarI and HuMSA-VAR, so annotated protein. It is composed by a number of, well, around 12,000 proteins and 100,000 variants. We actually generated, we had a lot, some missed protein while generating the embeddings with the protein language models. So the vectorial representation of the protein. So we have a slightly lower number, but this data set is basically derived from the ESNP and Go data set. And we also looked into a subset of what we produced, which is a subset of proteins which have at least one pathogenic and one benign variant. So you might have noticed that in the previous talk, they also mentioned the fact that sometimes concatenating several models, you might improve the performances. What we did instead was to look inside the embedding. So we delved into a single model. And to do that, we applied two different frameworks. One is the concatenation of the vector. So basically from vector generated from the protein language models together from the wild type protein and the variant protein, so like either benign or pathogenic. We would combine the two vector and come up with the concatenated vector, or instead we would do the difference between the two vectors, so wild type minus likely benign or likely pathogenic protein. And that would generate a completely new vector, which is the delta of these two vectors. Another thing that we wanted to look into was the different performances while we were looking into the vector that represent the full protein towards mean pooling. But also look into the per residue representation. This is a simplified version of what an embedding, or what a protein language model does. So it generates this vectorial representation of the proteins. So if you look into the per residue representation, you might think that looking into a window of residue is looking into a smaller piece of it. But if you don't do the mean pooling, you actually have much more features. So smaller portion doesn't mean smaller amount of feature. It always depends on how you look into it. So first of all, we started looking into what was the baseline of the ESNP and Go model. They used the ESM1B model, and they showed that just using that model with the concatenation of the two vector representing wild type and variant protein, the performances, okay, is this line, sorry for not highlighting it, were this, and we decided to consider this as the baseline, and we wanted to look into the new ESMC. We're actually not sure yet why the first test we did had such poor performances, but then when we tried with the delta, we noticed that the performances were quite comparable with the baseline. And but of course, the aim is to reach the level of the best performances showed in SNP and Go, and they have actually a huge amount of feature because they concatenated several models. Then we looked into the per window representation, and the per window representation showed us that the best window was the 3 residue window. So either concatenating or using the delta representation of the per window embedding, we have quite similar performances, very well comparable, but that means that the delta one is actually much smaller in terms of features. And this is just the overview of all the performances put together. As you can see, the 3 residue window is our best pick. We then wanted to look into the differences in the embedding space, and the 3rd residues window has already quite remarkable distances that can be measured and are visible in the embedding space, and the AUC based on the distances is already interesting and noticeable. So we see that there is a difference between benign and pathogenic variants in the embedding space. These are just some metrics on the subset we looked into, which is the subset with the protein for which we had both pathogenic and benign variants, or another version of the subset where there was just one benign or one pathogenic, and we actually got quite interesting performances in the smaller subset. And well, this slide I will just skip because I'm almost finishing. I don't have any more time. Well, to conclude, the best binary classifier reached an average ROC-AUG of 0.88. The classifier using the per residue embedding is slightly better, and but it's always important to consider if you want to use the delta or concatenate all the per residue representation, that would increase the computational resources you need. And finally, this decrease of performances that we observe evaluating the prediction of the subset of variants, exclusively pathogenic or benign variants that we noticed. But the concatenated representation in this case was slightly better. Finally, I would like to acknowledge the department, my supervisor, the colleagues of the biofold unit, and the Ministry of Italy that found my research. So thank you very much for listening.
Yeah, I think we have to skip because we are all late. So Marco will be around if you want to make questions.